Today we are here with Felicity Muth. She's an assistant professor of neurology, of neurobiology, physiology and behavior. She got her BS in zoology in the University of Edinburgh in the United Kingdom, her PhD in biology in the University of St. Andrews, also in the UK, and her postdoctoral research in the University of Arizona and University of Nevada. Her lab works on the cognitive ecology of wild foraging and lab-based bumblebees, focusing on cognitive abilities that have a clear function in bees' natural environment. Current projects in the labs include investigating cognition in relation to complex floral rewards and comparative cognition in wild bumblebees, and she also works on anthropogenic effect of bee behavior and cognition, in particular focuses on pesticide exposure. And today she's going to talk to us about how do animals think, some insight from bumblebees. So thank you so much, Felicity, and please go ahead. Thank you.
Yeah, thank you so much. And thanks for the opportunity to give this talk. It's a real privilege to be part of the campus community. And it was actually really fun putting this talk together because I decided to, I was thinking about the kind of broad diversity of scientific backgrounds of people who would be in the room. And so I decided to make my talk pretty kind of broad. And I also wanted to make a talk that would also be accessible to people who aren't scientists. And so I've kept my graphs to a minimum, a real minimum. And so I hope everybody, enjoys this talk. So before I get going, it looks like Earl maybe has his mic off. I thought I would just give a little bit of background of who I am. So I grew up in the United Kingdom in London. And then during my undergraduate degree, I moved up to Edinburgh. And that's where I first kind of discovered scientific research. I was working in a lab on these guys, this Nasonia parasitic wasp. And I fell in love with behavior. And so I then went to study animal behavior at the University of St. Andrews, which is just north of Edinburgh. And I worked on nest building behavior in birds, both with these southern masked weaver birds in Botswana and these zebra finches in mostly based in the lab. And after finishing my PhD, I knew that I loved animal behavior and I loved animal cognition, but I was ready to kind of have a change, both in terms of what I was working on, in terms of a system, and in terms of a scientific environment. So I decided to move to the USA. And so I moved first of all, to the University of Arizona. Here it's shown on an accurate map. And I started working with bumblebees. And this was in 2012. And I kind of haven't looked back since. I absolutely love working with the bumblebees. And so I then moved for postdoctoral positions at the University of Nevada, Reno. And there I did 3 different postdocs in the same lab, mentored by Annie Leonard. And I think I feel really lucky in that I've experienced a number of different labs, all with excellent mentors. And all with kind of different mentorship styles, which is something that's been really helpful for me in starting my own lab and thinking about different ways to be a mentor. But it was in Reno, Nevada that I also kind of discovered my love for the American West and doing field work with the bees out here. And so I started my lab, first of all, in 2019 at UT Austin, but continued doing field work out in Nevada and California. And then just last year, I moved to UC Davis. And so my lab is now here. And so this was my last lab photo taken at UT Austin just before I moved. But my lab has been quickly growing here at UC Davis. So here are my postdocs who started in the last few months. And while I've been trying to grow the lab slowly, we have quickly gained a lot of undergraduate researchers because it's just amazing how many students are enthusiastic and to work in labs and just seem like great students. So we've very quickly gained a lot of student researchers in the lab as well. And in addition to my kind of academic past, I've also really been interested in science communication for a long time. And so as a PhD student, I started a blog. This was kind of early stages, early times of internet blogging. And this blog then got picked up by Scientific American. I ended up writing this blog for Scientific American for a number of years. This blog was called Not Bad Science. And I also did some freelance articles here and there. And so science communication is something I've always kind of had alongside my academic career. And at a number of points in my career, I thought about kind of leaving academia and just doing science communication full time. But what I ultimately decided was that one of the best things about an academic career is you can kind of make it what you want it to be. And I've always been able to have the science communication as part of my academic career. And so I've done kind of interviews on and like discussions on NPR. I've done a bunch of different types of outreach event that I'll be talking about more in this talk. But I think that, right, especially in times like these, I think it's really important for scientists to be not only doing science, but also talking about the science that they're doing with lots of different people. Okay. And so broadly speaking, my lab works on cognition. And this word cognition, it is something that we use in our day-to-day speech, where it means a certain thing. But it's also a word that is used, I'd say, kind of differently in different groups of scientists as well. And so what I mean by cognition is basically everything that an animal perceives, how it perceives its world, how it processes this information, and then how it makes decisions based on this information. And this kind of definition is one that Sarah Shuttleworth, who was kind of a leader in the field of animal cognition, uses. And the reason we use this definition is because it's a very inclusive definition. It means that any animal that basically has neurons has cognition that we can study. It's not classifying animals as having cognition or not having cognition. It's kind of accepting that there's a continuum of cognition that we can study in animals. And so to study cognition, my PhD, I worked with birds, and for the last decade or so now, I've worked with bumblebees. And for bumblebees, they perceive all kinds of different stimuli as part of visiting flowers, different colors and scents and electrostatic fields. They have to learn which flowers have got the best rewards. And then they have to make fairly complex decisions between many, many different flowers in the environments in which they live. And bees, an individual bee can visit hundreds to thousands of flowers a day, and is constantly kind of integrating that information to make decisions to optimize its foraging. And in addition, a kind of theme of my lab that I'm not going to talk about today is also using cognition to understand how bees might respond to changes to their environment. So here's a photo taken nearby. Here we have, right, a wildflower strip on the left, but then agricultural land on the right. And we are obviously changing the environments of these animals incredibly rapidly. And so to understand how they might respond to that change, we also need a baseline understanding of their behavior and cognition. And it's not just me doing this. People have used bees, honeybees and bumblebees for a really long time to study cognition. So Carl von Frisch, back in the early 1900s, won the Nobel Prize for his work on honeybee cognition. And more recently, I would say in the last few decades, bumblebees have really, gained a lot of attention for their cognitive abilities. And so part of the reason is that bumblebees are really motivated and they will partake in a whole bunch of different tasks in a lab environment. So here is a bumblebee pushing at a red tab to get a sucrose reward. Here is a video of a bumblebee pulling at a piece of string to get at a sugar reward on this flower. And so bumblebees, they are very flexible in what they will kind of engage with. We can get them to engage in a whole bunch of different types of protocols in a lab environment. And using experiments like this, people have shown all kinds of different cognitive abilities. So improving on others, things that other bees have learned. People have done recent work on numerosity, so demonstrating that bees have a concept of zero and of basic addition and subtraction. There's been recent work on bees playing, and even, for example, the formation of abstract concepts, optimism, pessimism. It kind of feels like if somebody looks to see if bees have a particular cognitive ability, then they seem to find it. And one question I often get asked is why? Why are bees so smart? And I put smart in inverted commas here because this is a term that in animal cognition we kind of hate. Because just like, say, with physical abilities, there's many ways for an animal to have better physical abilities, right? That all depend on its environment and what it's trying to do. It might be better to be bigger, it might be better to be smaller, it might be better to be faster or slower or stronger in this particular muscle. And so the same is true for cognition. There are many, many different dimensions of cognition. And animals' cognitive abilities reflect selection from their environments. But for bees, I think, right, the reason that they are so good at engaging with many different types of tasks is because bumblebees are generalists. They visit many different types of flower. And in visiting so many different types, they have to rapidly integrate information in order to make the best decisions. And they're able to form kind of general rules about their worlds when they do this. And in my lab, we are mostly interested in understanding bee cognition in the context of this ecology. And so we do this both in our lab experiments, but also by looking at cognition in wild bees. And so today I'm going to talk about just one experiment that we did in my lab, because I think it's a nice example of how we can use bumblebees to think about processes that go beyond just bumblebees. And then I'm going to talk a bit about cognition in wild bees, because we have a number of projects going on right now relating to this. And then finally, I'm going to talk about bee diversity and some of the outreach that we've been doing. Okay, and so this first experiment I'm going to talk about, this was led by my former postdoc, Claire Hemingway. And this, the context for this experiment is really that animals have to make decisions. So this butterfly here has to decide whether it wants to oviposit on this leaf. This chimpanzee is deciding what food to eat. And this is a sage grouse, a male sage grouse, and females have to decide which male, are they going to mate with this male or not. But decisions like this aren't normally made in a vacuum. They're normally made in comparison to other things. And so this butterfly is deciding, should I oviposit on this leaf or perhaps on another plant species? This chimpanzee is choosing between many different types of food, and female sage grouse compare males to each other. And so we know that this is how many animals make decisions. The quality of an item is often viewed comparatively rather than in absolute terms. And so to give an example of one particular process within this, let's say that you're happy drinking this box wine. You're like, this wine is great. I drink it every day. I'm very happy with this box wine. You then try some fancy UC Davis wine, and this wine tastes amazing to you. And you then go home and you try your box wine again, and now this wine tastes bad. And so something that was previously fine now is viewed as not so good in comparison to something else that you've had. And this is what we would call negative incentive contrast. You also get positive incentive contrast, which is the opposite effect. But this process, this is found across, I would say all animals. I am not sure if that is true, but my gut is probably across all animals, including bees. And so for bees, for foragers, what they care about is floral reward quality, like nectar. And so nectar is largely, it has many different compounds in it, but it's largely sucrose, so sugar. So a bee might be perfectly happy visiting a flower with 30% sucrose, but then it encounters a flower with 50% sucrose, and this is amazing. And now when it samples that 30% sucrose, in a flower, it might have just been minutes or seconds ago that this was perfectly fine, but now that bee will reject that flower. Now this flower tastes disgusting to it. And we know that this incentive contrast effect is well documented in bees, and we know that it can happen with just the sugar alone, just with the sucrose alone. But what we wanted to know is how the features that go with that sucrose, so a flower, affect their perception of that sucrose. And so it put another way is this incentive contrast effect affected by the labels, the stimuli associated with that reward. And to give a human example of what this might look like, let's say we have our fancy wine in a bottle with a fancy label. We're then pairing that fancy label with that fancy wine. We have another wine that's got a basic label and it's lower quality wine. If we then take that fancy label and we stick it on that lower quality wine, then what happens? Do people view that as better or do they view it as worse? And what people have found in psychology studies where they do this kind of thing is that it can go either way. Sometimes we get a process called assimilation, where people will drink this and they'll say it tastes better because they're expecting it to taste better from that label. And sometimes you get the opposite effect, which is called contrast. And I should say that it's kind of confusing, but this contrast is different to the incentive contrast I've just described. But the idea behind this is that it tastes worse because we're expecting better. And so people have found both in this context, and we wanted to see what we would find with the bees. And so to do this, we designed an experiment where bees foraged on artificial flowers that were a particular color. And so in one treatment, all the flowers are blue and they have 50% sucrose. And then after 3 trials, in a final test phase, the bees are given flowers that have 30% sucrose. In our treatment 2, it's exactly the same, but we've just changed the color of the flowers. I'm calling this our familiar treatment because the colors are all the same. And then in our second 2 treatments, the bees are given exactly the same sucrose concentration, but we just flip the colors in that test phase. And so the bees experience a novel color or label or stimulus, whatever you want to call it. And so to do this, we use these bumblebees called Bombus impatiens. They are a bumblebee species that is native to the eastern side of the USA. And they are used in commercial pollination. So what's nice for us is we can buy these bees online. They come in a box. And then their colony, we keep them in lab and we can work with individual bees that we mark to then visit artificial flowers. And so in this experiment, we had a colony inside a colony box that we connected up to this foraging arena. And the bees were given these artificial flowers. So what they had is a colored disc that the bee could see, and then an Eppendorf tube that they would climb inside. And then this hole here is where I would pipette in, or Claire would pipette in sucrose. And so I'm going to show you what this behavior looks like in a couple of videos. So this is now the backside of that arena. And so, we can't see the color, but that's on the other side. And the bee is going to fly into this Eppendorf tube. And this is what she looks like. She comes in and her tongue comes out and she drinks that sucrose. And so we can really clearly see that she has accepted that sucrose reward. When a bee comes in and sticks out her tongue and rejects it, then she tastes it for a second and she's like, nope, I'm not going to have that. And so what we looked at was how much rejection the bees showed in that test phase. Because, right, like we're expecting rejection in all of these treatments because they've had this higher quality sucrose that's now dropped down. They're going to reject it. But what we wanted to see is how much rejection we would have in these familiar and novel treatments. Because if bees don't have label-based expectations, if the color doesn't matter at all, then we expect this incentive contrast, but we're not expecting any difference between these two treatments. But if this incentive contrast effect is affected by the labels or the associated stimuli, then we expect a difference between these two treatments. And depending on whether we see that assimilation or contrast that I mentioned earlier, it could go in either direction, as with our wine example. And so this is the slide I showed before. What did we find? Well, we found evidence for contrast. And so it tasted worse to the bees because they were expecting better. And so what we specifically found is when that label stayed the same, when the flowers went from 50% in blue to 30% in blue, the bees rejected that. They also rejected it in the novel color. But when we tested the bees over a period of time, they would continue to reject that familiar flower for longer. And this makes sense in terms of learning theory. The bees have learned, okay, blue flowers should be good, and now they're not. And so I will continue to reject this for as long as I remember that information. And so this finding by itself, it's not that we were the first people to find this. This has been shown in kind of classic experimental psychology studies with rats. But we were the first people to do it with a kind of a non-model species and thinking about it in an ecological context. And so what does this mean for bees? Well, what it means is if a bee is visiting flowers, it's visiting, many different flowers which have got different stimuli. Here I'm focusing on color, but really it could be anything. It could be scent, it could be other features. The implication is if it's already visited some flowers or one species, if it then approaches another species, it doesn't come with those prior expectations of what to find there, and it might be more likely to accept a lower quality reward. And many flowers of different species will do what we call facilitation, where they appear similar to each other, and they do this to enhance their pollination success. So plants will converge on similar phenotypes with the idea that bees will learn, okay, like these big yellow flowers are rewarding. I now visit this other species of big yellow flowers, looking for a reward there. And what our results indicate is if that second species doesn't have equivalently good rewards, then they're more likely to be discriminated against by the bees than a novel flower would be. Okay, so for the second part of this talk, I'm going to talk a bit about cognition in wild bumblebees. And before I get going, I'm going to ask everybody just to think of a bee. And I guess I've done some priming with lots of pictures of bumblebees. But really, when you ask people to think of bees, most people will think of a bee that looks like this. And so this is our European honeybee, of course. But there are thousands of other bees out there. We have around 20,000 species of native bee worldwide, 4,000 species in North America. Just within the bumblebees, we have 250 species worldwide and 50 species in North America. So there's this tremendous diversity. And even just within a single bumblebee colony, there are individuals that specialize in different roles. And so despite this tremendous diversity, most of what we know about bee cognition comes from these 3 commercial species that get used in the lab, like the species that I just talked about, and one particular life stage of the colony. Because when we think of bumblebees, what we're generally thinking of, say you go outside and you see a bunch of bumblebees and some flowers, these are foraging workers. And this is at a particular time in the bumblebee life cycle. But there's this whole other part of the bumblebee life cycle. And so basically, the life cycle includes these queens. They overwinter underground. They come out in the spring. And at this stage, the bumblebees they don't have a colony, and they're essentially solitary individuals. The queens then will start their colony, produce workers, and at that point, the queens then move into a reproductive phase where the workers go and collect all the food from the flowers, and the queens stay inside the colony. At the end of the colony life cycle, new queens are produced, males are produced, they leave, they mate with other queens and males, the colony dies, the males die, and then those mated queens overwinter and start the whole cycle again. And so I got interested in this stage of the life cycle, these foraging queens. And specifically their cognitive abilities, because these bees, these are these kind of absolutely massive bumblebees. They're often kind of 2 to 3 times the size of worker bees. And they're out at this really critical stage where everything is on this one individual. They can't rely on the colony structure. If they make a mistake and don't have enough food, that's the end of their fitness, right? Whereas the workers are part of a much bigger their genes are distributed across the colony. And so you might think at this stage, okay, there's going to be a lot of pressure for these bees to be really good at learning. And so this was something I was interested in looking at. And so I developed a really simple method for measuring cognition in wild bees, where we can catch a bee, put it in a tube and present it with different colored stimuli. So here's a bee where there's a reward, a sucrose reward on this blue strip of paper, and the yellow strip of paper is unrewarding. And we can basically catch a bee and train her very quickly that one color is rewarding, one color is not rewarding, and then release her again. So it's also nice in that this is a non-lethal technique. And just as a side note, this has also been a really useful teaching tool. It's a nice way for undergraduates to be able to work with, stinging insects in a contained environment. But also, the students can actually carry out a learning experiment in the course of kind of 30 minutes to an hour. So I've now done this in classes with over 100 students and 100 bees in tubes, where everyone is training their individual bee. And so it's a really nice way for students also just to get kind of hands-on experience with a learning experiment. Okay, so back to my experiment. Like I mentioned, I was really interested in queen cognitive abilities. And so during lockdown, actually, I went out and I collected bumblebees, this Bombus vosnesenskyii. This is, some of you might recognize this bee. It's a common species around this region. And I collected them from a couple sites across Nevada, California. And I found that, as I expected, that the bumblebee queens were really, really good at learning. They were better at learning than the workers. And, at the beginning of this talk, I talked about some of the amazing cognitive abilities that workers have. Foraging workers are capable of many impressive feats. But despite that, the queens were better at learning. And so this kind of led to the question of, okay, is this definitely learning, or is it something else? Like these queens, they are much bigger than the workers. They have bigger eyes. We know that bees with bigger eyes can see better. Is it possible that the bees can just see these colors better than the workers, and it's not actually learning, it's just perception? And so to follow up on this, I asked whether this learning performance was limited to the visual modality. And this last summer, I trained queens and workers to learn an olfactory association. So this is what this looks like. Here's a bumblebee being trained that a particular scent is rewarding. So on the first strip of paper was a scent, and the second strip of paper is a sucrose reward. The bee sticks out her tongue. She's like, that's great. I now give her a second scent, and these are floral scents. This one has water. She tastes it and she's like, nope, that's disgusting. I'm leaving. And so I can train a bee over, I think this experiment, I did 5 training trials. And then in the test trial, both scents are unrewarding. And so now these strips of paper have nothing on them. One is one scent, one is the other. You can clearly see the bee sticks her tongue out for one of these scents to show that's her choice. And what I found is that the bees learned the scents also better than the workers. So, okay, there's learning ability. It seems to not be constrained just to vision. It also seems that they can learn scents better. Okay, and so like I mentioned, these queens, they're really big. They have bigger eyes, but they also have bigger antennae. So we might also expect that maybe they can just smell things better. Bees smell through their antennae. And so then what I did is in this experiment, I took all the bees and I trained them up to make sure that they all had learned the same. And so this is what that data looks like. This is these bees were then given a number of more trials to bring them up to this kind of ceiling effect of learning. And then I dropped the scent concentration down. And this allowed me to test, are these differences driven by learning, or are they driven by an ability to just smell the scents better? And what I found was when I dropped the scent concentrations down, there was no difference between queens and workers. And at the lowest scent concentration, it does kind of look like the queens might be a bit better, but this doesn't come out statistically. And so no, there doesn't seem to be a difference in their ability to detect these scents, but it seems like these queens are just really good at learning. And this is something that we're now following up on in the lab under, more controlled conditions. Okay, and so bees can learn really well, but environments can change rapidly. So this bee might learn that this Wyethia species is the best thing in town, but a few days later, the Wyethia might be dead, and now a different plant species is the one that is rewarding. And so bees don't just have to learn associations, but they also have to learn to forget these associations and learn new things.
A little after 4.
And this is something people have thought about more broadly, about this trade-off between learning new information and then having to forget that information when there's potentially conflicting information. And so I put up an example here. I used to live in Edinburgh, and I lived in this house, and I did a great job of learning what my house was. But then I moved across the street, and I did such a good job of learning where my house was that I kept going back to my old house and trying to stick my new key into that door. And so sometimes if animals learn something really well, they then have this cost of when environments change, not being able to flexibly adjust to that change. And so this was something that my graduate student Smriti was interested in looking into, and she did this with help from Angie, who was an undergraduate in the lab at the time. And they used this species, vancouverensis, which is found here in California. And so Smriti wanted to know, okay, so these queens, they're really, really good at learning, but does this come at the cost of not being so flexible? When environments change, do they then have a harder time unlearning what they have learned and learn something new? And no, she found that the queens not only are better at learning, but they also reverse these learned associations better. So they can quickly forget what they have learned and learn something new. Another topic that we're really interested in the lab is queens themselves undergo these dramatic life shifts where they start in a foraging phase and then they switch into this reproductive phase in the dark, where they're never foraging again, and instead they're laying eggs and kind of having behavioral control of workers. And for this, we're looking into how their cognition changes between these two stages, and also how their brains change, specifically the morphology of their brains. Because we might expect, for example, that the visual regions that are so important during the foraging phase are no longer as important during the reproductive phase, whereas the olfactory regions we would expect would be very important in both phases because they're still using a lot of olfactory communication in their reproductive phase as well as the foraging phase. And so this is a project that's being led by my graduate student, Leah Richardson, in collaboration with two insect neuroscientists, because that is not my expertise. But here we have a bumblebee brain that has been stained, and Leah has been painstakingly calculating the volumes of these different brain regions. And this is work that, you know, we're continuing to work on in my lab right now. Okay. And so the story so far is that queen bumblebees seem to be really good at learning. And now we basically want to use this to understand more about how cognition could vary between queens and workers. My guess is that they're not just better at everything, that queens have specialized cognitive abilities. And that is something that we are trying to delve into at the moment. And we're doing this with a grant that I recently got from the National Geographic. And so this grant, it's a new initiative called the Wildlife Intelligence Project. And the purpose of the grant is to study cognition in wild animals. And so we're using this to learn more about these bumblebee queens. But in addition, like I mentioned, there are all these other species out there. And so we are both working with multiple species of bumblebee, but more broadly, we're interested in cognition in other native bees as well. And so part of the grant is also going to involve thinking about cognition in other native bees and how different cognitive traits evolve. And so these native bees vary in all kinds of ecological variables. So for example, whether they are solitary or social, whether they're generalists or specialists, and how these different ecological variables might drive variation in cognition. Okay, and so finally, I wanted to talk, I'm just going to check my time, yep, a little bit about the outreach that we've been doing in the lab. And so as I mentioned, when most people think of a bee, they think of the honeybee. And there's this tremendous diversity of native bees out there. But at the same time, the honeybee gets all the attention. And the honeybee gets all the attention because it's a bee that we use in agriculture. But the honeybee is not native to North America. It was brought over by European colonists. And yet when it comes to kind of saving the bees, the honeybee is the one that gets all the attention. And so this, you know, you might have heard of greenwashing. Within the bee community, this is called bee washing, where a lot of the initiatives that you see about saving bees are directed at the honeybee. And the honeybee isn't the one that needs saving. It's all the native bees are the ones that are in decline. And indeed, many of the native bees would actually be better in our agriculture than the honeybee. And so it's a bit like kind of like saying save the chicken when bird species are in decline. And so this is something that people in the media have been popularizing. You know, there have been articles like this: Move Over Honeybees, America's 4,000 native species need a day in the sun. The best way to help bees, don't become a beekeeper like I did. What we got wrong about saving the bees. And so, you know, I think that it's not that people aren't trying to talk about it, but when I chat to people, even scientists, right? I'm guessing that there's a lot of people here today that didn't know this, didn't know that honeybees are not the ones that we need to be worried about. And so it's great that people are writing about it, but it doesn't seem to have kind of bled into the general knowledge of the public. And so, you know, as researchers, you know, we're busy kind of doing our behavioral experiments with these bumblebees. And meanwhile, you know, we're reading articles like this about bee declines, popular articles like plummeting insect numbers threaten collapse of nature. And you start to feel a little bit like the dog in this meme where everything is on fire. And meanwhile, you're just there, you know, testing your bees in tubes, which I'm sure is also a feeling that many scientists can relate to right now. And so, kind of with all of these feelings, something I ended up doing was writing a children's book. This combination of knowledge about the massive insect declines that we are experiencing, and then combined with this kind of misdirected effort where there are people who they care, but their efforts are often misdirected, where people think, okay, bees are dying, I need to go and buy honeybees and keep bees, in my garden, for example. And actually, this can be doing more harm than good. And so what I decided was if I could at least teach children that there's more than one type of bee out there, this could potentially have a larger impact than the whole of my scientific career. And so I wrote this book called Am I Even a Bee? That is about this native bee, Osmia, that is a blue-green bee. And I'm not going to read you the whole book, but I thought I would share just a few pictures from it to give you the gist of what the book is about. And so Osmia is this bee that when she is young, she is told by her mother as an egg that she is a bee. But when she's grown up, she goes out into the world and she sees a lot of other bees and bees on people's t-shirts, bees in magazines, bees on television. And all these bees look the same, and none of these bees look like Osmia. And so she starts to have a bit of a crisis about whether she actually is a bee at all. And even some of the other insects, like the flies and the moths and the beetles, look like these other bees. And she sees these honeybees out being social, dancing with each other, but she lives alone in this little hole down here. And these bees, they look really different. They're round and they're fluffy and they're yellow and black, whereas she's blue and green. Her hair is patchy and she's slender. And at first, she tries to look more like these bees. She paints herself and she puts feathers on herself to make herself look fluffier, but it doesn't really work. And so she's feeling dejected about whether she is a bee at all. And she then flies into this carpenter bee. And she explains her situation to the carpenter bee. And the carpenter bee says, well, they call me a carpenter bee, but actually I'm terrible at building things. Maybe you should meet some of the other bees in your meadow, and you will realize that there are many different ways to be a bee. And so the Osmia then meets this sweat bee that's drinking cow's tears. She meets digger bees and parasitic bees. And at the end of the book, sings a song to celebrate the fact that there are many ways to be a bee. And so this book is, it obviously has it has this message about bee diversity. It also has a message that is pro-diversity more generally. Something that has been really rewarding to me is that I've been contacted by lots of different people saying, oh, this is a book about autism. This is a book about LGBT folk. And really, right, it's a book about diversity. And so I think it can kind of apply to anyone. And I think it's a message, right, that is very appealing to all of us. And so what I wanted to do with this book was both obviously celebrate diversity, celebrate the ways that while things like bees can seem different, ultimately we are all the same. And then at the same time, I wanted children to learn that there's, many ways to be a bee. And so in this book, one of the key things we wanted to do was make the bees look biologically realistic. And so here what I have is photographs of the actual bees on the right and the illustrations from the book on the left. And then on my website, the amievena.com, we have a lot of information for teachers and people who are trying to learn about these different bees in relation to the characters from the book. And this is something that we're going to be kind of building upon soon. Okay, and so this was at the book launch in 2022. And so something we have done is every time I do a book reading, we combine it with outreach activities. And so here are a bunch of scientists at that book launch. Here in my lab at an event that we did at a bookshop in Austin. And so here we have on the right, Alex Wild, the insect taxonomist put together a collection of the actual bees, and so children could pair, seeing the actual bees with the characters from the book. Most recently, we did an event at Texas State Museum, which had a really great turnout. And then just a couple weeks ago, we held an outreach event as part of Biodiversity Day in Davis. And so this book has been really handy in order to kind of act as an anchor text for outreach in schools and in libraries. It's been really nice to see reviews from librarians and from teachers online. And then something that's been great that has happened, which is what I was really hoping, is that other groups are picking up the book as well. So this pollinator nonprofit that in Oregon contacted me and sent me these lovely pictures showing that they've been using the book to educate kids about bees as well. And so something that I am doing now is I'm working with a teacher in the area to basically make these, I have some bullet points of facts about bees, but these are obviously not super engaging educational materials for kids. So I'm working with a teacher who's an expert in this to develop educational materials that can be partnered with the book. So we involve like the characters from the book in the educational materials. And so just to finish, I guess, by saying that, this feeling that I think we often all have of the world being on fire whilst we're trying to kind of continue with our science. For me, I found that one of the ways that to deal with this is to focus on a particular thing that is important and that you can really, contribute to. And for me, just knowing that some children are learning more about the insect and bee diversity that is out there has been really helpful. Okay, and so with that, I will thank my lab, my funding, and take any questions.
Thank you so much, Felicity. It was an awesome talk. Congratulations. So please, questions.
Hi, Felicity. It was a really great talk. Thank you. I have a really quick question about memory. So there was a lot of testing going on to show that learning is taking place. But I wonder if your studies have also included how long that acquisition of new knowledge sticks with the bee. Yeah, I'm just a little bit curious about that.
Yeah, so some of my past work looked specifically at bees learning about pollen on flowers. And in that study, I found that individuals could remember the information for about a week, which is kind of pretty long for individuals that only live for a few weeks. Something that we are interested in doing in my lab is, the foragers are only alive for a few weeks, whereas the queens have this much, they have an annual life cycle. And so looking at, the queens and how long they remember information for.
Some more questions, please.
So I'm not sure if I understood the point that you made about the bees that were when you like the knockoff flowers that maybe aren't as sucrose rich, that they recognize that those knockoff flowers aren't as rewarding in terms of the return that the bee gets, and the bee recognizes that that the knockoff doesn't have the same quality of sucrose as the first one, like it was like the two yellow flowers, right? So does that mean then the bee is not likely to would recognize a knockoff and then is unlikely to return to that knockoff flower?
Well, so I guess the main point of that is that, we often think about how well, so I guess there are 2 points. One is that people have shown that response, that comparative incentive contrast response of, oh, this was fine a second ago and now I don't like it. That isn't just related to sensory processing. There's also a learned component that goes with it. And then the second key thing in terms of what I think what you were driving at, like what does that actually mean for a bee visiting flowers? And what does it mean then for the flowers? Because, right, this is a co-evolved system, is that if a bee is visiting one flower and then another flower that is similar, it's going to have higher expectations for that second flower. And so in terms of that flower and its evolution, if you're going to mimic another flower, then you better have the rewards to back it up.
Yeah.
Yeah, so
Sorry,
Nelson,
one
really,
quick question. Am I still sharing my screen?
yes.
Yes.
How do it's weird when I click on share, it asks me to share my screen again rather than to stop sharing. I don't know if
It's a nice picture. That's
okay. All right.
we can still see you, although you're small. Hey, great talk.
Thank you.
Lot in it. I'm going to ask a kind of open-ended and probably confused question. But you focus a lot on the reward side of learning. And obviously wild bees and a lot of wild animals, I think more about birds, think a lot about the cost side of foraging as well. And it's got to be a big deal in these big meadows. You're a small bee, and it seems like a kilometer away. It seems like a long ways to go. And then that comes into sort of spatial learning in addition to sort of color or olfaction. Bees obviously learn and remember locations in a large environment. And I'm wondering if you're, when you're thinking about the neural correlates that you mentioned that you were working on, whether you were thinking about whether you measure spatial learning in the different stages of life in your bees, and B, whether you're anticipating any changes in the sort of spatial parts of the bee's brain, which are big. I mean, there's the whole torus and I don't know, all parts that help them navigate.
Yeah, no, I think
Go
that's a
anywhere with
great
that.
point. Yeah, I think that we don't like bees pay attention to spatial information both at very small scales and at really big scales. At the small scales, we control for it. And while I would love to study it at the big scales, we haven't yet just because of the practical limitations, right? The bees can fly for like miles and learn.
Yeah.
And I would love to, I'd like dream of the day when we can stick little radio tags onto bees and see where they're going.
You can. No, can't the MODIS tags go down to that scale?
No.
They do it on butterflies.
Well, what kind of technology is that?
It's a little RFID thing that can get very, very small.
We can do
MOT
RFID tags, but then you need to have the readers out for them
Right,
to
you need a
go
big
through.
antenna array.
Yeah.
I don't know the process of it. Again, with birds more than bees, but
I think you're right in terms of right, the bumblebee queens go from having this very large world to this very small world.
Mhm.
And so you might expect that that would be a part of their brain that they're no longer needing to use.
Yeah.
More questions?
I had kind of a question, I guess I don't know if you know the answer, but with the incentive contrast, how many pairings with that higher reward do you think is necessary to achieve the incentive contrast?
Yeah, it happens incredibly quickly. I think one pairing.
Oh, wow. Okay.
Yeah.
Okay, more questions? I think that we are done then, Felicity. Thank you so much. This was a really awesome talk and very comprehensive. So thank you for making it easy for us. And see you in 2 weeks, everybody. Thank you, Felicity.
Yeah, thank you.
Thank you.